Right, it's my great pleasure today to introduce Dr. Mathieu Wimmer. He's an assistant professor at the Department of Psychology and Neuroscience at Temple University. Welcome, Matt.
Thank you. Thank you for having me. It's great to be here.
Great. So Matt and I have known each other for some time. We actually initially first met because of our lack of overlap in the same lab. So he was I believe, undergraduate, if I'm not mistaken, in Gary Aston Jones's lab at the University of Pennsylvania at the time. And he worked on and was an author on this Nature paper showing the role of orexin in reward for one of the first times. And this was a really important paper for my own career. So it was kind of a pleasure to meet him a couple of years later through mutual friends and things. And I've been paying attention to his science ever since, and I'm really excited to talk to him today. So we're going to be discussing a paper that he recently published with collaborators in his group in psychopharmacology. It's called Parental Morphine Self-Administration Produces Object Recognition Memory Deficits in Female but Not Male Offspring. And this is by Alexandra Ellis et al. Out of the Wimmer lab. So Matt, could you tell us a little bit about the main findings of this paper?
Yeah, sure. So I guess to set it up a little bit first, one of the questions that really drove the project was to think about, there's a lot we know about what drugs of abuse do to the brain and to behaviors in general. And so there's all these comorbidities that sort of arise with chronic drug use in addition to, the substance use disorder itself. And those sort of include changes in mood, changes in sleep, and also changes in cognition. And so a lot of that work has been really well defined, particularly when it comes to psychostimulants and somewhat with opiates and opioids. But the general message here is that chronic drug use can lead to cognitive deficits. I'm thinking particularly in the domains of memory and attention. And it's really interesting because they both can result from the drug use itself, but they're also thought to potentially contribute to the cycle of addiction. And some of the evidence for that is that cognitive deficits related to drug use are typically predictive of treatment, retention, and outcomes, right? And so this observation, several decades ago at this point, has led to the thought that perhaps a better understanding of these cognitive deficits and targeting them with therapeutics could be a new avenue to either both treat substance use disorders and also understand them. So that's sort of like the ethos of the question. And then in our models, we're also interested in knowing, does that actually happen across generations, right? We know that within an individual, it's very likely that chronic drug use can cause a lot of these things to happen. But we know much less about how experiences in either the maternal or paternal lineage can influence the next generation. And as you know, that's becoming more and more relevant with the ongoing opioid epidemic and more individuals consuming prior to conception and babies being born either from mothers who are using drugs during pregnancy and thereafter, and or dads. So that was basically the idea.
Yeah. So would you say then that the premise here is that obviously these addictive disorders tend to run in families many times, and I guess the obvious assumption would be that there's some genetic predisposition that could put people at risk of addiction. And so what you're saying is that maybe it's not just the genetic predisposition, it's actually also things that the parents do themselves that could change essentially their genome or their at least epigenome. Is that kind of what the premise is here?
That's exactly right. And so we know that risk and a susceptibility for substance use disorders and other psychiatric disorders is really rooted in a complex interaction, as you just pointed out, of genetic and environmental factors. And so the rats that we use are outbred rats. So yes, they're somewhat genetically diverse. So they're not clones of each other, but they're also not as genetically heterogeneous as you would find in the human population, certainly, or even the heterogeneous stocks that have been produced for these kinds of genetic studies. So yeah, you're right. We're really focusing on the environmental influences, particularly the drug exposure in the parental generations, and trying to ask, does that matter for the next generation? And if yes, potentially how is some of that information transmitted? And actually, I'm glad you brought this up because we really focus on the paternal lineage for two reasons, I would say. One is that it really gives us a way to probe the biology behind the potential transmission, and I can explain a little bit more why as we keep talking here. But it was also kind of an open question in the field. There's been many more studies on maternal manipulations than paternal manipulations, and if one of the questions is about the transmission, then actually studying sires or dads is a little bit easier. Because they don't contribute typically in a laboratory setting in raising the offspring. And we already know from a wealth of literature that maternal behavior and maternal care can impinge upon a lot of these behavioral endpoints we're interested in when we try to model psychiatric illnesses in rodents. So knowing that maternal care isn't going to be disrupted by the drug use itself, potential withdrawal effects, as well as removing the possibility of in utero exposure. If all we care about, if one of the things we care about is the transmission, that makes our life a lot easier. Now, I'm certainly not saying that the maternal studies aren't important. They're extremely important. I'm basically saying they're very complex and difficult to interpret at times. And so because one of our questions was about the transmission, we decided to go with the paternal lineage.
No, that makes a ton of sense. I mean, it's the idea there, I guess, is that you have, essentially the male sperm is changing in some way that then you can uniquely identify what are those changes in the sort of germ cell that then somehow influence behavior, et cetera, in the offspring, totally excluding anything that could have to do with beyond that sperm. So like basically any kind of maternal care, et cetera, is not there in a male. So you're really just isolating this really interesting transgenerational inheritance phenomenon, right?
Exactly.
Very cool. So what'd you find? So what'd you do and what'd you find?
And then actually in this article, we also sort of validated or delineated the details of our intergenerational model. So let me tell you a little bit about that. The males have access to morphine self-administration daily for 3 hours. And we do this for 60 days, 60 plus days. The reason for that timeline is that that's roughly the duration of spermatogenesis in rats. So that's the period of time during which sperm cells are being produced. And it's also the window of time during which all the machinery that can remodel the epigenetic landscape in the germ cells is present, active, and vulnerable to potential insults and outside influences. So the idea is that a substantial proportion of the mature germ cells, by the time we pair those drug-exposed sires with drug-naive dams, have been produced in the presence of morphine in this case. And then so you have the parental generation where dad is exposed to morphine or saline, and then mom is never exposed to anything. And then the first generation or F1 generation is what we focused on in this article. And we asked a lot of questions. We asked about anxiety-like behaviors, and we asked about memory. And we also asked about the HPA axis. So we found, actually encouragingly, we found very little differences in anxiety-like behavior. So we used two different tests. One is called the Novelty Induced Hypophagia Test, or NIH, often used for short. So in this test, the animals can eat peanut butter chips in their home cage, and we sort of train them to do that. We just train loosely here, but we acclimate them to eating the peanut butter chips. And so once they get over the initial neophobia, and once they actually taste the peanut butter chips, they realize they love them and it's safe to eat them in the home cage. And then we changed the rules on the rats and put them in a anxiety-producing environment. It's a new environment, it has different smells. It's also open and brightly lit, which is anxiety-producing. And so now the rats are conflicted between wanting to eat the peanut butter chips and also staying away from predators and staying out of the open area of this novel environment. And so one of the measures that's really useful in this test is to ask how long it takes the rats to approach and eat the peanut butter chips. When they are in their home cage, they do this, and they've been acclimated, they do this right away, within seconds. When we put them in this novel environment, they don't do it right away. And so we see a huge increase in the latency to consume. And so this happens in both our controls and our morphine-treated offspring. Morphine
so the longer they wait, the more anxious they are, basically.
Exactly. And so, you know, it's a nice, within subject control, we know that there's going to be a huge spike in the latency for every individual, but that was not affected by siring. So our interpretation was that anxiety-like behaviors were not affected by paternal sire history, paternal morphine history, excuse me. And so we did another test. So that's an appetitive test. We did a probe burying test where the animals are come into contact with a shock probe and then bury the probe as a result. This is, again, an innate behavior. And we kind of know from a wealth of studies that the longer they bury the probe, it's likely that they're more anxious, and drugs that tend to reduce anxiety tend to also reduce the burying time. And there was no difference there either. So, okay, no differences in anxiety-like behaviors, no differences in the HPA axis response. But when we started looking at memory, that's when we found interesting results. So we used both hippocampus-dependent and hippocampus-independent tasks, and found that hippocampal function seemed to be intact. And so for those two tasks, we used contextual fear conditioning, as well as object location memory. And so these objects, I really am a big fan of these object tasks because they actually don't produce a lot of anxiety. They don't heavily rely on motor behavior or a lot of other functions that can be considered as confounding factors. So their performance on the task essentially is not likely to be altered by other non-specific effects. So we found that, again, the hippocampus-dependent tasks were fine in both males and females of morphine-sired progeny. But when we looked at object recognition, which engages the perirhinal cortex, that task was actually impaired at a 24-hour time point and only in our female morphine-sired progeny. So that was really interesting because a lot of this intergenerational drug exposure work tends to produce sex-specific phenotypes in the next generation. But many, many times, it's the males that are actually affected. And so this is the first study that I've done, certainly, where we saw a change in only the females and not the males.
Interesting. So is this, is it then fair to wonder if this is like a, so I guess the difference between a sperm that will end up with a male offspring and female offspring is what's in the sex chromosome, right? So is it a Y or an X? Is that relevant here? Is this like the mechanism, part of the mechanism? You think this is an X-linked thing, or is that too simplistic? Or could you say more about that topic? I'm curious.
I definitely don't think it's too simplistic, but so far in all of the work we've done to probe that question, we haven't found an easy answer. You're right that there are X's and Y sperm, and it turns out it's somewhat different. First of all, I want to say that I'm not a developmental biologist. I've had to learn a lot about this space because of this work, but
me too. We can pose together, I guess.
All right, but I do. We collaborate with a lot of folks who are experts in assessing the sperm genome and epigenome, and they tell me that it's pretty difficult to separate them. To me, that would be an important first step in trying to analyze them separately. And we don't think a lot of these effects are driven by mutations, genomic mutations. And the reason I say that is that typically you would have a very heterogeneous phenotype. And so the fact that we're able to pick up pretty consistently these differences in populations that emanate from different litters and different dads suggests that it's probably at the epigenetic level, right? So thinking about how the DNA is packaged, when it comes to the germline, one of the components of the epigenetic landscape that has garnered a lot of interest are the small non-coding RNA. And so Tracy Bale and others have done a lot of work in this space, and Ali Rando, to try to better understand how these small non-coding RNAs are even making it into the zygote in the first place. That's kind of a big question. But also, how are they remodeled and reprogrammed by experiences of the dads before they conceive? And so it's really an exciting space, I think, because of all the work that they've done to sort of lay the foundation. And, you know, as neuroscientists and running experiments in the lab all the time, it's really exciting to know that there's a potential functional experiment at the end of that tunnel, right? Where you can actually inject pronuclear zygotes with a small RNA cocktail so, you know, getting back to what you were saying earlier, defining those germline signatures, I think is one of the exciting potential future directions of this project. So once, if I've convinced you that there are systematic and reproducible, reliable changes in behavior that are caused by the paternal history, right, paternal drug history, then I want to understand how that's being transmitted. And is that consistent across cohorts and across cohorts, right? So can we define that signature? Can we point to specific aspects of the signature that are going to ultimately influence the neural development of these offspring, right?
Absolutely, yeah. And of course, then not just are they consistent across cohorts, but the real actions is this like happening in humans, right? Like, is it really true that the like sort of literally sins of our fathers are, causing us to make certain decisions or like sort of have certain kinds of psychological features or memory or whatever? I mean, that's hugely important. I mean, this is basically sort of, essentially like a non-Darwinian view on genetic influences on behavior in some ways. Like things that you do can influence not just you and your lifetime experiences, but even that can be passed on. So what's your take on that? Like how much is this happening all the time? Like everything that a father does will impact their offspring, or is it sort of specific to drugs, or is there, what do you think about that kind of stuff?
Yeah, I mean, I do think, I think there's evidence to show that the environmental influences certainly change the content, for instance, of those small non-coding RNAs of the sperm. So I think the biology definitely responds quickly to the environment. Whether or not that ultimately translates into drastic changes in behavior in humans, I think that remains highly debated. And that's not exactly the question we're trying to answer. I think what I'm hoping could translate out of our findings is more on the biology side, right? Can we identify, specific small non-coding RNAs that are conserved that tend to respond to opiates more so than stress, right? There's a stress signature, there's an opiate signature, maybe there's diet signature, things like that. Kind of thinking about it the other way where, because one of the other things I haven't mentioned explicitly is that what's exciting about the potential to translate some of this work is that the biological material is readily accessible, right? It's not invasive. I mean, one of our collaborators, we talk about sperm a lot in the lab, so there's always jokes. And so one of them said, if you want to, build the study, they will come. And so there you go. We've never done that, but again, I know this is work that Tracy and her collaborators are really pursuing and actually making headway, and others in the research triangle are also doing a lot of this work. So I think if we can find some parallels across species at that level, then we can really start asking real questions about what are those consequences in early developmental stages in both species, and then we can get somewhere. But for me to sit here and say, you know, opiates are going to cause cognitive deficits only in female children, I think doesn't make any sense. You know, it's just too different of a model.
Yeah, it occurs to me that you know, part of this could be sort of similar to how you're approaching this question, because looking at sperm-based inheritance of these sorts of things is just more feasible, more easy to do in a concrete and provable way. Yeah, it seems like you know, reducing the experiences of these animals to like nothing really ever happened in their life, so to speak, except for like they had morphine for 60 days. That's like a huge signal on a really small background. And so perhaps like reducing the system in this way to prove sort of that this can happen is an effective way to do that. Now, is this like likely to have massive effects in humans? Perhaps not, because we have lots of experiences all the time that could be, for example, reversing these by, you know, getting better after taking drugs or whatever it is. But still, the mechanism is there, and we still need to know about it. It could actually be useful for us therapeutically as well, I assume, right?
I think that is the idea. And don't forget, you know, there's several rounds of erasure of the entire epigenetic landscape as the germ cells mature. And so, you know, there are systems in place to make sure that a lot of these signals aren't actually transmitted. And so I think you're right, that is what becomes interested, is what does remain, what does make it through, and how salient is it, right? I think you hit the nail on the head there. From an evolutionary perspective, you better make sure this signal is salient if it's going to be transmitted. Otherwise, you know, it doesn't seem to have much of a point.
Right, and I wonder then, is this sort of like a brain thing? Because I mean, like the classic example is like the Lamarck, the giraffe. You know, the leaves are high, the giraffe stretches to get them, and then over generations, the neck gets longer and longer because of this parental stretching or whatever. So can you imagine that these sorts of things, maybe even drug experience itself, has also effects on like peripheral genetic processes of any kind? Or is this like something special about the brain that allows this to sort of influence stuff even in the next generation?
Yeah, I think that's a great question, and I don't have a great answer. But what I can tell you is what we've observed, which is that specific domains of behavior seem to be particularly affected. A lot of times that's sex specific. And likewise, at the molecular level, the signatures of different brain regions are completely divergent based on the paternal history. And so, you know, you have to also remember, I guess I didn't explicitly say that we are studying adult offspring, right? So they're 60 plus days old. So a lot has happened by the time we get to that brain. And I think, you know, it's a question people ask all the time, and I think it's a great question, but it's really difficult there are just so many steps between our insult and what we're doing that, and I also don't have the expertise, to be perfectly frank, to really, you know, delineate each and every single one of these steps. But I think starting with that signature in the germline and early developmental stages is really the key. And then I think once you have that end of the picture, then perhaps we can start asking more questions about the adult brain. But, you know, one thing that we've tried to do, you know, so we're not going to answer all of these questions, but one thing that we certainly are excited about is that using this model can systematically produce offspring that are going to do one thing or the other, right? So you know what the adult brain is going to end up looking like or behaving like in some reliable ways. And I think using that as a way to ask more questions about, you know, what makes a brain vulnerable to addiction or vulnerable to depression and things of that nature. I mean, that's been a question we've asked as a field for decades, and it's really difficult, right? It's anytime you study something, anytime you expose the animal to a drug or a stress, you've already changed it, right? So to categorize animals into these subpopulations is really tricky without changing it. And in this case, what I'm arguing, I guess, is that we can have this really early life insult, and we can sort of predict which bucket the rats are going to fall into, and then we can try to better understand those buckets. So that's sort of like where, at least where we are with these projects in my lab. And then maybe work our way back one day with the help of a lot of collaborators.
Absolutely, yeah. No, it's never as simple as it seems, and obviously, like what you guys are working on and what you, I think, demonstrated in this paper is more like proof of concept. Like this is something that can happen. Now they're associated. What is the mechanism? Is it that this epigenetic change somehow influences like responses to novelty, and that's why they learn different, or is it that it influences? For example, you also are studying maternal care and how that can impact the developing brain. So can it change the response to maternal care or incorrect maternal care or something like that? And then it has the secondary effects. Like there's a lot of possibilities here, and obviously a whole career or more than that to really map them all out. But you got to start with the phenomenon, and I think that's where you guys are really making some important headway at the moment.
Absolutely. And the exciting thing about developmental aspects is that it provides windows of potential intervention, which is, I know, something, the clinical field is desperate for, right? With what do you do with children that are born, with withdrawal syndrome? And what is the best way to treat the mother and the child, and what is going to lead to the best outcomes? Things like that can actually make a real difference, in the near future. So.
Absolutely. And I think also just, if you have a negative outcome you want to treat, and in fact, there's like two groups of people that fall into that category of negative outcome, like two different depressed people, basically, one with a problem stemming from something that their parents did or something that happened early in their life, and another that came to that same phenotype a different way. The therapeutic, strategies may well be different. And those are the kinds of things I think you also are honing in on with hopes for future treatments that are more precise.
The other question we really haven't touched yet, but I think we're really excited about is how long, once you have defined, let's say you do define the signature, how long does it last, right? It matters what dad is doing, but how long does that matter for? Will it be rectified? And I say that both because I think that's an inherently interesting question and something certainly people ask a lot after our talks and things, but also we view it as a strategy to hone in on the parts and the aspects of the epigenetic landscape that may be changed. So what I'm trying to say specifically is if we wait, let's say we wait 90 days, right? Wait another 4 rounds of spermatogenesis, and then breed the animals again, we have found that any phenotypes that we find in the first generation tend to go away, okay, for the most part. So if that's true, then we can actually compare the early versus late signature of morphine-exposed dads, if you will. And whatever remains, we know it's probably not, functionally relevant. But so that added level of comparison, in addition to the staining group, I think is a great opportunity to delve further into those potential mechanisms too. So that's some of the things that we're working on now, but we don't, we haven't made much progress yet.
Yeah, absolutely. I guess one of the implications of what you just said might be that if, there are these transgenerational problems that come from drug use, maybe you could just stop using drugs and have a kid later and you'll be okay. Or maybe you're sort of marked for life, so to speak. Like that's a big difference. And, I think you'd want to know, so glad you guys are doing those studies as well.
Exactly.
Yeah. And great. And then just one small technical question I was just curious about is, why did you guys pick morphine of all the drugs? Sometimes people have like, heroin's sort of a famous opioid, but it's also Schedule 1, so it's like inconvenient to use. This is sort of behind the scenes kind of thing, but these are the decisions I think we scientists are making all the time. Like, why this model, not that model, when there's not necessarily a strong theoretical reason to choose one or the other. How did you come about that decision?
Yeah, it's a great question. I mean, the truth is that morphine was what we had access to when I was starting this work, and we didn't have access to heroin. But I also think, so I guess, is it a model of addiction or is it a model of pain management? I'm not sure. I don't think it's really either necessarily. I think it may have relevance for, there's certainly morphine is still used widely in the clinic. And so there's a fairly broad number of people who are exposed to morphine pretty chronically and routinely. So I think it has value there. But at the end of the day, I think it really just gets back to what is the question, which is, do opiates ultimately, have these long-lasting effects across generations? And I also wanted to say, all of the work we've done is intergenerational or multi-generational. We cannot call it transgenerational because that really requires that the affected generation didn't have a direct exposure to the insult. So on the paternal line, that would be in effect in the F2 generation. So if we took the F1s, bred them to drug-naive females, and then the phenotype is still preserved, then we can call that inheritance transgenerational because it went all the way to a generation that wasn't directly exposed to the morphine.
Gotcha. Thank you for correcting me. That's shows my naivete in this kind of thing, but it's an important point because, like, let's say there's just a bunch of morphine in the semen, for example, that could have effects of its own that have nothing to do with the sperm. Theoretically, probably not. But yeah, thank you for that question. Great point. Yeah, any other last comments you'd like to make, or other things we didn't get a chance to touch on yet?
Well, I appreciate your having me on, and it's been fun to talk to you about it. I think, again, I think a lot of this work we were trying to do to answer some questions that have been harder to answer in the field using a very specific model that we can keep consistent and be reproducible. I think a thing that's really hard with a lot of these studies, there's a lot of skepticism, rightfully so, and we really need to convince ourselves that the phenotypes are real and that we're and reproducible before we really get into the mechanisms. So it's long and hard work. I did want to acknowledge Lexi Ellis, who was my first tech and really spearheaded these studies. And it's a lot of hard work. But I think at the end of the day, it has the potential to answer some questions that we've had a hard time addressing. So that's my hope. And we'll see what the next steps are.
Well, hugely important work, and sometimes the hardest questions are the most important ones. So keep it up, and I'm sure we'll be learning much more about this, and we'll be watching your future papers to hear more. And just one more time, I just want to thank you again, Dr. Wimmer, for your time and this really fascinating conversation on this extremely important and interesting topic.
It's my pleasure. Thanks so much. Thank you so much for having me.
Okay, cheers, Matt.